So we're going to start on the session on searching for extraterrestrial genomes. And what I want, when Drew, Andy, and I put this together, we wanted to put together a session that sort of demonstrated the wonderful complexity and commonalities of life as we know it, mainly microbial life, the most highly evolved life on Earth. And so we're going to have 3 talks on that. One talk on new forms of life that can be designed, another talk on the genomics of intelligence, and another one on how messages might be present in genomes. So I'm going to start with sort of why I believe, as you saw in this voting, if any of you noticed, there were 2 hands up that were sort of strict panspermists. One of them was mine. And so it's, one was Pete Warden, so yeah, that's good. Yeah, I hate panspermia. I hate panspermia. It would be a much, is that, you need to, no, lateral transfer of life is fine. Okay, so let's go on. Let's put on the slides. Am I up? Yeah. So this is a project that I've been interested in with Chris Carr, Mike Finney, and Maria Zuber, which is to look for life on other planets that looks like life on Earth, and I want to rationalize to you why I think it's a reasonable thing to do. And the title here is that what is true for coli on Earth would be true for life on Proxima Centauri, and that's a warp of the Jacques Monod quote, where he was asserting that if you learn something about E. coli, you've learned a lot about how an elephant works. And it's really about the unity of life on Earth. So this has been launched in a big way by genomics. And you know, genomics was originally just let's pull out a gene and see what the structure of a gene was. And it sort of happened in my scientific lifetime to sort of watch the genome revolution and to be a small part of that. And these days, if you look in the Today section there on my slide, there's thousands of eukaryotes, that's organisms with chromosomes and nuclei, but tens of thousands, hundreds of thousands of bacteria and archaea whose genomes have been sequenced. And yeah, the Genome Project for humans was, you know, a $3 billion affair, but that's early days, and now human genomes are a few $100s. And even my rather humble genomics lab, I'm not a big time, you know, $50 million lab. It's sort of a $2 million a year lab. We do hundreds of genome sequences a year now, routinely. And we're just emblematic of what thousands of genomicists do. So genomics has taken off, and now we can compare genomes in sophisticated ways. And this is an old figure from the fantastic textbooks, Brock, The Biology of Microorganisms, which I recommend anybody read. I've done 2 reviews on Amazon Books, and one of them was for Brock. It's one of my favorite books. And so these were the early genome sequences, and you can see the number of base pairs in a genome in that column 2 there. And there's sort of 1 million base pairs in a genome. And this was sort of my first experience of having 7 significant figures in biology, right? We finally had caught up with physics. My favorite moment was when I sent an email to Paul Horowitz showing lining up of ribosomal genes. This is in 1995, and I crashed his computer. When a biologist could crash an astronomer's computer, we had arrived. And he was mad at me, and I was going, yes, I crashed your. So notice E. coli is sort of the perfect organism that we know the most about on Earth, probably, and that's down that list with 4,600,000, et cetera. And to the right of that, you can see it says there are exactly 4,288 genes. So how do we know what are the genes? Well, part of that is by translating, this is what the E. coli genome looks like. It's a circle, right? So it's got the 4,000 base pairs in a circle, and you can translate them Here we go. So the ribosome that you'll hear about from Harry Noller takes the DNA sequence and does a transform on it to grab it 3 bases at a time and translate it into amino acids. And so you can look at the 4 million base pairs, and just in a computer, you can say, oh, look, I can see where all these genes are. And so the beautiful thing about the genomes of bacteria is, oops, I'm sorry, I hit the wrong thing. Here we go. Is that they have operons. And so you can see genes with names like ATPC, ATPH, ATPF. Those are all involved in ATP synthase, which you'll hear a lot about from Karen Davies. This is one of the most miraculous parts of all genomes. And so you can find them, and now that we have all these genomes, you can take the protein sequence of a genome. For example, there's a very short protein that's involved in the C subunit of ATP synthase. It's only 89 amino acids long, and you can see that it's conserved all across phylogeny, that all different bacteria have it, we have it, every organism on Earth has it. And what does it do? Well, in our case, these are the subunits that are involved in passing electrons from high redox potential in the metabolism. They pass electrons down their redox gradient, end up pumping protons across a membrane, and this is a very intricate machine. This is a highly evolved machine. Every bacteria has it. We have it. It's what makes our mitochondria work. It's why I'm moving on the stage. It's something that's been at the base of life, and it's really sort of how it all works. Now, if you look across genomes, you can find about 500 genes that are shared essentially across everything. That's the basic set of what it takes to be alive. And some of those genes, this is a comparison where we're lining up a query that comes from a worm and comparing it to an archaea. And you can see that there's amino acid positions that are unchanged. Everywhere where it lines up, those amino acid positions are unchanged over 4 billion years. So this protein was already evolved to the point where it works so well that it can't change in half of its positions since 4 billion years, right, at the base of the tree. So it's not just that life kind of got up to kind of working. It's like it got to being super highly evolved very fast. That's the problem. That's why panspermia is so attractive, is you either have to believe that, yeah, it's really easy to go from the RNA world to the DNA world, or you believe it just came here. And that's why I'm such a religious fanatic about panspermia, more so than most. So we have built a tool with Yuval Tabach where we take the score that we get out of these, what's called a blast, and so you can take that score and you can ask, blue means a high score, and so here's a gene that happens to be super highly conserved all across animals, plants, bacteria, and archaea. It's essentially solid blue all along. So that's like one of 500 genes that are conserved across all organisms. So if you go through and look at all the genome sequences, and again, 10,000 different bacteria, what are the sort of the common features? Well, yeah, there's a bunch of common conserved genes, but there's also a common theme that what all of these organisms are doing are taking a reductant, passing those electrons down a redox chain to an oxidant. That oxygen happens to be oxygen for us, but for other organisms, it's sulfur, it's iron, there's all kinds of redox gradients that are used, and all of them work by charging up that membrane like a little battery with a proton gradient, and then releasing that proton gradient on this miraculous motor that you'll hear about in Karen Davies' talk, ATP synthase, which is a perfect little motor that rotates inside of us. All of us have this little motor rotating. It's fucking great, right? And so this is like the core of all of biology. It runs it. And when did that evolve? Well, all of these organisms have it, and so it's at the base of the tree. So where do we have fossils? Well, we have, lots of trilobites and dinosaurs, but that's all up at the top. Up at the top of the tree here. So our timeline is sort of inferred from the rate of mutations. We don't have the timeline to the bottom of the tree. But we do have signatures, and so that's why the number 3.5 billion years is used a lot for sort of when the first isotopic evidence of life has been found. But plate tectonics erases history. Plate tectonics is really bad for doing history. It's good for the Earth. So yeah, the oldest fossil is 3.5 billion years, but you sure would like to look in older things, and it's hard to find things that are older than that. And that's one of the beauties of going to Mars, is that they don't have plate tectonics on Mars, and so it doesn't erase history as well. So if panspermia is a real thing, then Mars might have been seeded with the same life that we had, and they haven't been Mars hasn't been erasing history in the same way that Earth has. So that's one advantage of looking there. So the usual dogma that again, if you had a room full of biologists, this is a room full of mainly astronomers, if you had a room full of biologists, the voting would have been very similar, that most biologists would say, oh yeah, the RNA world was on Earth. And so the dogma is, yeah, we start with the RNA world, and you eventually get to cells, they're RNA-based cells, and then DNA somehow, the ribosome is invented, and that displaces it. And I do not doubt that any of this happened. I just personally doubt that the RNA, that it happened on Earth. And so that makes the evolution of life not an Earth problem, it makes it a galaxy problem, and that makes it about 10 to the 11th times more interesting as far as I'm concerned. So, okay, so the problem is, that it all had to happen fast, right? And so, and the other problem is directed panspermia. So there's two kinds of panspermia. There's the undirected, just things, it's a biological phenomenon, and microbes come here and they make the atmosphere. But we're going to do directed panspermia if we're really serious about colonizing planets. The first thing you're going to do to go to Mars is to direct a terraforming of Mars. And so 1,000 years from now, if we actually did this, within 1,000 light years, there would be panspermia that was directed by us. So in that sense, directed panspermia, it's a little more theological, because it's sort of like SETI. It demands, omniscient, cognizant beings. All right, so yeah, so isotopic fractionation is the method that is used. So enzymes work faster on lighter isotopes, just a mass effect. And so that's how you can see evidence of an enzyme in ancient sediments. And that's where the 3.5 billion year number comes from. And so one way to look for more ancient life is to look for isotopic fractionation of essentially modern life in older and older sediments. Okay. So sometimes when I talk about life on Mars to non-scientists, they say, well, who cares if you find bacteria on Mars? Because, why do I care about bacteria? But again, bacteria are the greatest things. So, people who are interested in robotics, a bacteria can make a copy of itself in 20 minutes. It's like 4,000, it's the most perfect robot ever, right? It's 4,000 gene products, and in 20 minutes, those 4,000 gene products can duplicate themselves and make another copy of that bacteria. It's stunning, right? So the idea that bacteria could go to a planet and colonize it is not crazy, and they do things, chemolithotrophic bacteria, all they live on, you don't need to give them anything organic. They don't need precursors to DNA. They're not sucking DNA from the environment. They're making DNA themselves, right? You give them a redox gradient, sulfur, you know, a gas to breathe, a little bit of CO2 to fix, and they can do it. So chemolithotropes, which are organisms, will grow on you know, just inorganic compounds. They generate that proton gradient, and all they need is a little CO2 for carbon fixation. They do it. And of course, the most perfect organisms for this is the photosynthetic organisms, not the plants. That's a late adaptation. But the photosynthetic organisms that do it without oxygen. They don't make oxygen. They use a different reductant. And they're using light, and there's plenty of light in any planetary system. So the simplest way to look for life these days is with genomics, and it's done a lot. And the best example of that is an Oxford nanopore, you know, which is 87 grams. It sort of sticks on your iPhone. There's thousands of people who sequence DNA. They do it in sort of wild places. They go to the Amazon. They go to places where Ebola virus is, you know, they use it for a lot of things. There's thousands of people who use these things. So if you sent an Oxford nanopore to Mars, you could look for DNA on Mars. So asking, is panspermia true between Earth and Mars? It's sort of, you can be skeptical about panspermia, but the idea that life would have spread between the two planets closest to each other, that's not so crazy, but it's sort of 10 to the minus 11th is interesting as spreading across the galaxy in my mind. But it's the first test of it, and we've already heard today that there can be good exchange between Earth and Mars. And it seems really idiotic to not look for DNA on Mars. If you say, oh, well, it could be different, you should at least look for the easiest thing to look for that's the most likely thing to find. And you can take advantage of the trillion-dollar genomics industry. All right, so I've told you about it. So this is how an Oxford nanopore works. We didn't invent this. This is invented by the nanopore people, and it's a E. coli protein that's actually involved in how E. coli spits proteins out of the cell, and it's fused to a DNA helicase, and so when DNA goes through that Oxford nanopore, it changes the conductivity of that thing, and so you can detect DNA by that. It is not the method of choice if you're a big-time genomicist. It's not as accurate as an Illumina machine. The Illumina machines weigh, 500 pounds. You know, you're not going to send a 500-pound machine to Mars. I put the market cap on here to tell you that sort of Illumina is sort of a 50th of Apple. So, you know, it's not the biggest company on Earth, but it tells you that genomics is you know, 2% of your iPhone in terms of what it means. So I think that's big. And so what would happen if you do a DNA sequence? The prediction is you're not just going to find DNA. It's not anonymous, yeah, we'll get a sequence. It's that you'll get a sequence that you can interpret. It's informational. You'll say, oh, it's deeply branching. It's related to life on Earth, but it comes from a deeper branch than life on Earth. Assuming that this tree of life is based on 4 billion years, right? What if the tree of life is actually a tree that started 14 billion years ago? So then it would fit into the tree with everything else on Earth. But it's an experiment that's worth doing, we would say. Okay. So in favor of panspermia, am I out of time?
Couple more minutes.
Yep, I got like 3 more slides. So what, you know, all the hot Jupiters and Neptunes, this is really talking about spewing of material between planets and planetary systems. Yes, it's hard to capture a planet, but it doesn't have to get captured if you're talking about microbial transfer. It just has to spew out some microbes that can be caught. It's like catching a cold on the train. You know, you don't have to kiss the person next to you. It just happens. It's the germ theory, which I believe in. All right. let me stop here. Oh, for the SETI people, I wanted to put this in. So if DNA sequences are universal, if they're sort of shared across planetary systems, you shouldn't be looking for your SETI people to send you pi and E. You should be looking for them sending DNA sequences. And if you look at the data on the internet, genomics is taking up a fair bit of the internet. You know, movies have displaced them now, but there's a lot of genomic data being sent. And so if I was to send out genome data, I would be sending out. And so you look for a symbol like that. That's who we are. Thanks very much. Yep. I think they turn it off for feedback.
Can you?
Yes.
Okay, that sounds better. Thank you for that intriguing talk. Something you said that really struck me is that a bacterium reproduces itself in 20 minutes. So there's something I think about from time to time as an astronomer called the Fermi paradox, which is, well, if there's someone else out there, why haven't they sent a self-replicating probe to go check up on us? And then it occurred to me, what if we are this self-replicating probe? Do you think that this is a good idea?
Well, so Drew Endy is going to talk about if there's directed panspermia, then what's sent out, wouldn't you think there'd be some embedded message in it? So we have all these genome sequences. Can we find embedded messages? I looked for Elvis years ago, because in the protein code, ELVIS, and I did find Elvis, but...
Right, but for instance, with CRISPR, today we could create that experiment. The flip side, we could take some bacterium and code something in it with CRISPR and send that out into the universe.
Yep, easy to do. It's just, these things mutate, so you need to have an error-correcting code, but I wouldn't put it past some clever person to do that.
One more here.
Hi. If you had interstellar panspermia, and that was the reason we have life on Earth, and obviously it started very soon after the Earth cooled, then wouldn't that be happening all the time, and would you have evidence for it in the genomic record?
In other words, would you see constant receding?
Yeah.
Yeah, I mean, you know, once it takes off, of course, you're sort of buried by what's here. But I think looking for sort of a bacterial flux, you know, I mean, that's the one use of the ISS I could think of for science. It's pretty useless scientifically, but it does have that one use.
Maybe one more comment, and then you'll introduce Harry.
Yes.
So a few years ago, there was a lot of talk about the functional part of the DNA sequence, and then this historical development of complexity in the DNA sequence over the evolution of life on the Earth. And at least two people made an argument that you could see a kind of a Moore's law in the evolution of functional number of base pairs over the species evolution.
I think it's an inverse Moore's law. So humans have a lot of junk DNA in comparison to these race car bacteria, but the number of genes is not that much more.
So you don't, my question was whether you had a comment on the timescales for the apparent information content or complexity that's measured by the emergence of a species and its number of functional base pairs.
Yeah, I mean, we're seeing very, very efficiently trimmed down genomes of 2 million to 4 million bases for different bacteria and archaea, and then humans go up to 3 billion years because we're really good at feeding, and so efficiency is just not a big issue for us.